Kia ora and welcome to our May LINKOnline webinar. We usually like to do these kind of in person, but for some reason the technology is not allowing us to share our cameras today. So we're sorry about that, but the most important thing is that you can see the slides and you can hear us. My name is Christine Harper and I'm the Stakeholder Relationship Manager at Manaki Whenua. I love working alongside our scientists to share their research, and we hope this will inspire you to think about what we're learning could be useful in your own work. Graham Hickling and Patrick Garvey are the joint leaders of a large research program called Eradication Science, which explores how we can eliminate the cleverest rogue pest animals from the landscape. Graham is a wildlife ecologist with a special interest in disease management, and Patrick has been doing some really interesting work to understand how animals respond to the different kind of scent cues they encounter. It's early days for this 5-year program, but already there are some exciting ideas to share, and I'll hand over to them shortly. I'll be back at the end to help with the Q&A session at the end of the presentation. Get your questions into the chat box in plenty of time. We might not get through all the questions in our live session, but rest assured we will follow up on all of them and publish responses alongside the webinar on our website. Now over to you, Graham.
Kia ora. Good morning, everyone. Thanks for listening in. So, as Christine said, we are at the early stages of a 5-year research program. And just to give you a little bit of background to how that program developed, Landcare and many agencies in New Zealand have a long history of tackling the problem of introduced mammals. And essentially that problem is that we brought in possums, mustelids, rats to an environment where there's an abundant food supply and there's a lack of the natural population controls, the predation and the disease that would regulate those kinds of populations. And so consequently, we have very high population densities of predators in New Zealand that then are very destructive. And so for years, there has been research and management action focused on dealing with large population numbers over large areas. And so New Zealand is a world leader in developing this kind of large-scale control program, where the target has been to get the majority of the pests controlled in a cost-effective way and over large areas. And so there's a lot of research we could talk about on that front, but the focus today is that after that kind of large-scale control, there are always some survivors. And the point that I want to make in the next few slides is that even a small number of those rogue animals can be very destructive. And so some quick examples. Here's one from Australia, where a single fox managed to wipe out 20% of the local penguin population in just a couple of weeks before they managed to tackle the fox and get rid of it. There's a very famous example in the early days of European settlement of New Zealand. So the Stevens Island Ren was essentially driven to extinction in just a couple of years. The story goes that it was the lighthouse keeper's cat, possibly named Tibbles. I make that point just because there is a certain amount of mythology around this story. What we do know is that a pregnant cat was released onto the island. It and its offspring seem to have essentially wiped out the Stevens Island Ren in such a short space of time that it was essentially extinct before it had even been properly described by science. There's much more modern examples as well. There was a male cat that was found to have killed at least 100 short-tailed bats. There was a German shepherd that had killed more than estimated 500 kiwi up in Waitangi forests. There was a single rat suspected of killing 80% of the shore plovers on Mana Island, and then another rat that did the same on Waikawa Island a few years later. So these individual rogue animals are very potentially destructive. And the other part of the equation is that they are also potentially very difficult to control, more difficult than the average animal in a population. And so one kind of interesting experimental example of that was a study done by James Russell and his colleagues on the Noises Islands, where they deliberately let a single rat go. The rat was radio collared, to watch what happened when a single rat arrives on an island. And it took them 18 weeks. Ooh, what have I said there? 18 weeks, not months. Sorry about that. But it took 18 weeks before they were able to capture it again. And when they did, it was in a trap baited with penguin meat, located in a spot that sniffer dogs had concluded was a high rat activity area. And over that 18 weeks, they had gone through a whole litany of different approaches. They'd tried live traps and snap traps, tracking tunnels. They had dogs out there. They had various kinds of bait, and they had various kinds of poison. And it wasn't until that, extended effort that they managed to get that one single rat. And as with the cat and the wrens, if the rat was pregnant when it arrived, it's not going to be just one rat for long. It's going to seed a new population. Another more recent example, a single stoat penetrated Orokonui Eco Sanctuary on the Otago Peninsula. Took 12 months of effort to get that one stoat. And in that time, it had managed to wipe out the local population of saddlebags. So the point we're making here is that after the large-scale operations, when you have the small number of survivors, or in an area that you feel you've succeeded in eradication, but then there's a recolonization event, these rogue individuals, these recalcitrant individuals, are really important for us to be able to tackle if we're truly going to move into an eradication mode for pests in New Zealand. And so that point's kind of summed up in this graph, that the early parts of a predator control operation goes reasonably smoothly, but as you get towards those recalcitrant animals at the end, the cost per predator, the cost to take out each of those predators, essentially starts going exponential. And so we've often heard about the 80-20 rule in lots of different walks of life, that you get 80% of the benefit from 20% of the effort, and then getting the rest gets much harder. And here we've got kind of a 95-5% rule. So to target that last 5% of the animals, the residual ones in a population, may well take you 95% of the cost and the effort. So that's the motivation, the philosophy behind the Eradication Science program. And so as Christine introduced, this was a 5-year proposal that we put forward to MBIE, and that was successful. It started running in October 2019, and then COVID hit. So when Patrick tells you about progress to date, keep in mind that there were a few hiccups early on. The program will run through to late 2024. The two key questions that we're targeting are, firstly, what are the behavioural characteristics of these surviving animals, the recalcitrant, the rogues? How are they different from the average animal? And then with that knowledge, can we use that better understanding to tackle them better? Are there cues? Are there combinations of cues? Are there traps, designs, and techniques that can get these non-average animals? And so the program itself focuses on 3 targets: the possums, stoats, and the 2 European rat species. Obviously, implications, the kinds of learnings we get from this expand into some other pest areas as well. The design of the program, we've split it into 4 research aims. And the first of those is to understand the behavior of the recalcitrant animals better. The second is to then manipulate that behavior to find better ways of targeting them. The third is to use Mātauranga Māori knowledge to inform and broaden the kind of approaches that we might be considering. And then finally, in the second half of the program, we want to scale that up into demonstrating we can actually apply this at a practical level, directed towards eradication rather than sustained control. And these 4 research aims obviously feed into each other. The understanding behavior feeds into manipulating behavior. The Mātauranga Māori insights will also inform that. And then both RA 1.2 and 1.3 will feed into the scaling up phase. So it is early days. We're only partway through year 2. And so we're going to focus the rest of the presentation today on just RA 1.1 and 1.2. And I'm going to pass over to Patrick to talk about those two research aims.
Thanks very much, Graham, and thanks everybody for joining. So what I'd like to talk about is the research we're undertaking in 1.1, and this is focused on understanding the behaviour of survivors. Who are these individuals, these 5% that are not being controlled, that are the most difficult to control, and trying to get some insight to who these are. So our key question in this area is what behavioural characteristics contribute to some individuals surviving control? And again, we're looking at these three species, primarily stoats, possum and shiprats. And our control methods, we're looking at both trapping and toxic bait. And the reason for this is because these two different control methods can influence the characteristics of the individual animals can influence their susceptibility to these two control methods differently. So we're looking at both of them separately. And then our central hypothesis here is the behavior traits. So each animal has behavior traits that are akin to personality, and these are consistent differences, individual differences that are present through time. So that's our key hypothesis. We also look at fear. So these might be innate fears of control devices of whatever that may be, and it might be neophobia or fear of novelty. We looked at learned avoidance, basically whether animals have learned specific things that will allow them to survive, such as if an animal got a partial bait and it was sublethally poisoned, maybe this is why that they are not susceptible to poisoning in future. And then we have a number of additional hypotheses as well. And these are other things that we believe might be happening, so that it could be intrinsic differences of the individual. It could be dietary preferences or microbiome. There could be genetic reasons for it, and there's also genetic markers related to personality, or indeed survivors could just be a random subset of the population. As we control individuals, we're reducing down and the random subset, and there's density effects because of that, and maybe that's why we're looking at survivors. So I'd just like to talk about these additional hypotheses first. So if we think about the intrinsic difference of the animal, what we're talking about here is the age, sex or condition and characteristics of the individual. Sorry, Graham, just on to the next slide. So these could be relatable to a number of different factors. So like a large male might have a certain behavioral response to a device than a small female or whatever that may be. So we're looking at it. What we're doing is we're looking at the general population. So we collect a sample of the whole individuals within a population, and we then compare that to the survivors, which is a small subset of this population. And we look at these intrinsic differences to see if they're consistent. We're also looking at diet and microbiome, and this can be linked to personality. Specialization or generalization can be linked to individual differences. What we're doing is, I won't go into it in much detail, but where we're capturing survivors, we take a small scat sample that will give you an indication of the food that was consumed in the previous couple of days. And we can also look at its microbiome, and that's a very interesting element of the research. We can look at whether there's microbial resistance to toxins such as 1080, and we're collaborating with colleagues in Australia to compare the microbiome of New Zealand possums to those that are found on the west and east coast of Australia, because there is high genetic resistance in west coast populations. We're also taking genetic samples from all the animals. There is some indication that there might be genetic resistance within a population. So all survivors that we collect, we take ear clipping and blood samples from each of these, and this is potentially going to be looked at by Otago students, and then just documenting whether survivors are indeed just a random subset of the population, and we're doing this as part of field trials. So when we think about personality or behavior traits, these are individual differences that are consistent across time. And there's a number of different traits. There's some described as the big five. And I'll just mention a few different traits here and how we're measuring these. So if we look on the left-hand side, we can see exploration and activity. And these traits, you can imagine, would influence whether an individual animal encounters a bait or trap, how active it is in its environment. And here we've Kyla, who's this is the bookcase. So what happens here is there's 4 different levels. The animal is introduced at the bottom right-hand corner. You record its activity for 5 minutes. Food items are placed on the different levels. And what the test is how much it used the level it least like and compared to how much it used the overall, the different levels of the boxes. Now, Kyla has worked in Australia on possum and rats and is currently running these possums, both survivors possums and general population animals, and running tests on these. Neophobia also could be a very important factor. If an animal first encounters a bait or trap, what's its response to this? And some animals innately will be neophobic and will avoid a device or a bait when first encountered. And you can test this. You can see the little children's toy there in the bottom left-hand corner, and there's the cameras on the roof of the cage. And this possum is asleep in bed. When he wakes up tomorrow morning, he'll wander out and see this unusual object. And we're seeing a range of different behaviors when they're first responding to this. And then we're linking these behaviors to how they interact with traps and device. Are they truly neophobic? And a final example is what we call the release test. This can, or the wriggle test, and these can tell you behaviors such as boldness and docility. So this can affect management in that if an animal comes up to a trap, a bold animal might be more likely to push through and interact with a device, where a more shy or docile animal might be less likely to interact. So these are the type of personality measures we'll be making and that we'll be linking across to the management tools. So when we think of survivors, it might relate to the fear of baits or devices. So pests are under selection pressure to recognize and avoid devices. You can imagine management is akin to a predator, and there's evolutionary pressure on individuals to recognize devices. So as animals are taken out of the population, the remainder they might have innate risk of devices, or they might be neophobic, as previously discussed. Or they might have learned. So this is a key element of the research as well. So if an animal escaped from a trap, or if it was sublethally dosed by poison, as people know, these individuals are nearly impossible to remove afterwards. So we're looking at whether these are innate, whether there's learned fears, and if they are, the second RA will look at how we overcome these fears once they have been established. So this is our first field study, which recently got underway. The field site is a lottery bush in Waiu, and what the site is around 150 hectares. Within the site, what we're doing is the area hasn't been subject to any recent control. So we go in there, we take a sample of the population as our general population animals. We bring them back and we run a series of tests and related to both feeding and response to devices and personality tests. Then the local community group undertake intensive trapping operation in this area. There's over 100 devices out, a number of different traps and different baits are used. And then we go in after the population has been knocked down to very low numbers, catch the surviving individuals and look to see if these survivors have different personalities than the general population that we saw at the start. We're using camera traps to see interaction with devices and whether they're avoiding it. And this should give us an insight into whether these survivors are true survivors and whether they have specific traits that make them survivors. And how do you catch animals that are impossible to catch? Well, we're going to be using things like leg hold traps that are not visible to the animal, and potentially dogs as well, to track down these final survivors. And survivors of trapping operations, the sort of questions we've been asking is how do pest responds to novel traps. So this can be done in captivity, and we expose them to a range of traps. Will they get captured? And what are the traits of the recalcitrants? So these are all things we're measuring captivity. You can see the possum on the left obviously had no problem going into a trap. But for the animals that don't, we're developing smart devices with our collaborators, Cacophony, and you can see there that they have the device there in the ground, and this can tell the difference between individual animals. It's automatically can be set off, triggered. An animal can look through the trap, so there's no fear. So obviously sometimes an animal is fearful because of the device itself, so that's minimized risk. There can be sound cues or scent cues used in association with this, and they're also using IR thermal cameras which can identify individual species, and this can decide whether it triggers and not triggers for non-targets. So this is developing research at the moment. So first I'd like to talk about a toxic trial that was undertaken last week. This was with a PhD student, Mel Bird from the University of Canterbury, and Alyssa Cameron. So again, we look at an uncontrolled population. We had 100 individual animals that we bring into captivity that have not been subject to control. And then we went into an area that had very recently had a dock operation, and went in and put a lot of effort into try and capture survivors. And what we're looking at is, again, our measures such as personality, neophobia, how they respond to different food items, how they respond to an example of the cereal bait that was used in the operation. And we're also looking at diet and microbiome. And with the uncontrolled population, we look at their personality, we expose them to 1080 in captivity, and we look at who survives and see if these personality can be linked across our neophobia, or indeed their diet can be linked across to help explain these survivors. And just a very quick video here, you can see that bait in the center of the screen. The possum first rocks up and he brushes it aside, kicks it away. He doesn't like that at all. And this is an animal that was captured from an area that had been 1080 controlled, and then we tested these on a bait, a cereal bait with no 1080 that was the same bait matrix as that, and bait scent, a cinnamon bait that was used in the 1080 operation. And this can give us an indication, are they really true survivors of the operation? And of the 16 animals that came in, 12 did not touch the bait, 2 of them chewed the bait and then rejected it, and only 2 animals out of 16 ate the bait. So it gives us a clear indication that some of these survived the control operation. So this is a second big study that's taking place this winter. I'm very excited about this study. So again, it's taking place in Franz Joseph. It's a dock operation that will take place in September, and we're looking at survivors, the average possums from the population versus survivors. So again, the focus is cats or rats and possums. So what we're doing is going into the area, capturing over 60 possums and 60 rats each, bringing them back to captivity in land care, doing a series of tests, personality, genetic and diet. And then we're bringing them back to the area again, putting GPS collars on these animals and releasing them, and then looking at who survives the trapping operation and whether this is relatable to personality, diet or genetics. Again, the species are possums or rats, and there's Grant with a GPS collared rat. So one really interesting aspect of this is the GPS collars can tell us when the animal died. So not only are survivors important, but also at what point in time, how far into the operations did the animal survive, and if this is relatable to the measures that we took. So just briefly, I'll talk about research aim 1.2, and this is manipulating the behavior. We know there are individuals that survive, we understand who they are, and how do we overcome these survivors, these behaviors that allow them to survive. So again, how do we use sensory cues, and how do we alter perception of risk to improve captures of these animals? When we talk about sensory cues, what we're focusing on is sound, sight and scent. And for scent and sound and sight, what we're basing these on are the primary motivations of all animals. And these are called the 4 F's of behaviour. And these are sensory cues that we can use to target the pest motivations. On the right-hand side panels, you can see what the 4 F's are: fighting, fleeing, feeding, and fornication. And these are the primary drivers of life. And we will use these 4 F's to help us focus on sensory cues that will be appropriate for these animals. And we're also thinking about how do we change perception of devices, where these individuals are fearful of devices. How do we overcome this? And John Dyer sent me that photograph of a modified dog trap. It looks very interesting as a change, looks a bit different from the norm. And simple adjustments like this can overcome. And also we're working very closely with Cacophony on their smart devices, where these will overcome fear because the fear of the device has been removed. So that's what we're developing, a device that will minimize the fear associated with trapping. And just briefly, the next steps. So very excited to get results are coming back from Mel very shortly. And so we'll know about these survivors of 1080 and the link to personality. We're looking at behavioral responses to devices at the moment with possums, rats, and stoats in captivity. The 2 big field trials, Lottery Bush, got underway last week. They caught 35 possums in the first few days, which is great. The 1080 field trial will take place in September. And then RA 1.2, this has been led by Al Glenn. We'll send trials. We've started in collaboration with Mike Jackson from University of Canterbury, testing 4 F's scent cues and also 15 food lures. Auditory trials have taken place with stoats and more to come. And we're doing some visual lure trials, which Al Glenn will lead in the coming months. And then we look at cues in combination instead of just singly. And also we look at automatic devices with Cacophony, and these smart devices are under development and getting better all the time. So this is a very collaborative, very large-scale project. It's Lancaster Research with funding from MB. We work with DOC, Predator Free 2050, the National Science Challenge ZIP, and Hawke's Bay Council. Vision Maturanga is a key element of this research. I didn't talk about it today, but that's RA 1.3, and we should be able to give you an update very soon on that. And that's a key element, and the Māori researchers are driving this element of the research. We're looking at, we're working, our sensory cue research has been done with various universities and Cacophony. And then our personality-based research is being done with UC Davis, again, University of Sydney, and again, our other collaborators, both in New Zealand and abroad. So thank you very much for listening, and I look forward to your questions.
Right. Thank you very much, Patrick and Graham. That has been fantastic. It's been so fantastic that we've pretty much filled up all of our time. And I'm aware that busy people have things to go on to. So rather than even trying to tackle one of the great questions that have come in, we will respond directly to those people who have asked the questions, and we will also be posting the Q&As on our website alongside the recording of the presentation and the slides. Can we just go to the next slide, please? There's a whole lot more research going on across the biosecurity spectrum at Manaki Whenua, and we have a Biosecurity Bonanza, 8 webinars over a week's period that you can sign up for to hear more about our research in this area. So if you go to Manaki Whenua's main website, search for Biosecurity Bonanza, and you'll be able to sign up there. So I'd like to sign off for now and say thank you very much. It's great to see so many people here, and thanks to Graham and Patrick for a really fascinating presentation. Bye for now.